Well, thank you all for coming back on time, and we'll continue with several presentations on the same theme that we've started this morning. And our next speaker is Dr. Adelina Vuchkova from Yale University. Adelina received her PhD at Yale and stayed on and been a great collaborator of Dr. Anastas, and he already gave her a good introduction, and she told us that all the details are in her talk, so we're looking forward to that.
On.
It is on.
Yeah, great. I don't know. Great. Well, thank you so much for the introduction. And I'll just start off by saying that this morning we heard a lot of great talks on the state-of-the-art methods for toxicity assessment. And what I hope to do in the next half hour is show you a little bit about how we can not only use this data to get direct information about the hazards and toxicity of chemicals, but how we can hopefully learn from this data and understand how we can rationally think about designing some of that hazard out of the chemical. So it's a real challenge currently. And of course, what I'm saying is directly stated in one of the principles of green chemistry that Paul put up as an eye chart earlier. And of course, our goal is not only to have a chemical that has the desired function, but also one that has minimal hazard. And that is something that is done at the design stage. So this is the key here. And so if we think about what needs to happen for this goal to be realized, I think we can appreciate certainly from this audience, that not only do we need an understanding of the chemistry of the molecule, but we certainly need the toxicology and also the environmental science or ecology. So these three fields really need to come together for us to be reasonably able to achieve this goal. And before going further, I think it's important to take a step back and look at where this goal is today, this subfield, let's call it, of green chemistry. If we look to the left of the slide, we have to acknowledge some of the key founders that have, inspired this field. And at the bottom, I start. Whoops, my apologies. With E. Arians, that in the 1980s put forward some ideas in some of his founding papers that showed, from a medicinal chemistry point of view, how it is possible to start thinking about hazard minimization. And some of the other familiar names that you'll see here, certainly John Warner, Paul Anastas, but also some key colleagues from the EPA, such as Dr. Steve DeVito and Robert Bothling, have been working in this field for a number of years and trying to push it forward and inspire it. From industry, we've seen today, and also from several examples of winners of the Presidential Green Chemistry Awards, examples where these principles and this methodology has been put into practice, and we've seen the design of safer chemicals and safer products. But if we look at academia as a field and look at the papers that have been published in the Journal of Green Chemistry over the past 10 or so years, it would be very hard to find a paper or two on directly molecular design for hazard minimization. And we did this assessment when I first started working with Paul about 3 years ago, and we could find one paper that dealt with this. And of course, the papers that deal with toxicity are, again, very few, 2 or 3 over the course of the last 10 years or so. So this is really a nascent field, and it's exciting for someone starting out like myself to be working in this field. But certainly an opportunity for many other groups from academia to contribute. So let's think about why there's so little that's being done. I think that there's a good reason. This is one of my favorite wall charts, and it's not an eye chart for reading, certainly, but the Roche Biochemical Pathways chart is a summary of what we know about biochemical pathways, and I think we would all agree that there's a lot we don't know that's not captured here. But the complexity of this chart, I think, is really mind-boggling. And if one imagines that, our goal as chemists, from what I stated, would be to try to understand how to avoid hitting all of these pathways, we can begin to appreciate the challenge that we're really facing. And of course, if we think about some industry members that have tried to do this. I think our colleagues from pharma would show us that there are ways we can design chemicals to hit specific pathways. But again, we're looking at trying to avoid hitting all of these pathways. And so I've just put a comparison list here of what some of the characteristics of pharmaceutical and industrial chemicals are, where, of course, we're trying to design all biological activity out of an industrial chemical. And we're looking at very diverse use scenarios of these chemicals, whereas pharma is really concentrating on very specific use scenarios of their target drugs. So I think a lot of potential cooperativity between these two fields, and a lot that we can certainly learn from the great strides that pharma has taken. So let's try to simplify this. And is it really possible to come up with some rational design guidelines from what we know, despite the complexity that I just showed you in that chart? And I hope to show you that there really is. So the way that we started thinking about this about 3 years ago was to imagine what the gates of chemical exposure would be. And we saw a beautiful chart earlier this morning from Dr. Tangwei that showed points of entry of a chemical into a set of biological pathways. Well, similarly, one can think of a chemical that has to become bioavailable to exert a toxic effect. So that's, our first sort of fence. The second would be that it has to become distributed to a certain extent to exert its toxicity. The third might be that it needs to either be bioactivated or eliminated. So if we try to reduce bioactivation to a more toxic species or increase deactivation, then we have a potential defense. And finally, we have a variety of very specific target interactions that typically are associated with toxicity pathways, and talking about receptor-mediated pathways, interactions with DNA that ultimately lead to the toxicodynamic interaction. And so we can target that interaction as well. So with these layers, I'm going to talk in these 4 points about what we currently know that we can use to stop a chemical from interacting with the body in these 4 stages. And then I'll talk more generally about how we can design general guidelines. Before doing that, I have to show you what the key properties that we're going to use are. We use various software packages, and we're developing our own for calculating properties that are relevant to toxicity and biological activity. On this pyramid, I hope it's visible, we're starting from size and shape and going all the way to phase interactions. And of course, size and shape in electronics are associated with intramolecular interactions, while solubility and phase interactions deal more with intermolecular interactions. But you see some of these listed here. Electronics, for example, include dipole moment, the energies of the frontier orbitals that chemists can calculate quite easily, polar surface area, and all the solubilities and partition coefficients that are associated with biological activity. So all of these are known to influence various biological effects, and certainly biochemical interactions such as Herc channel, blockage, and so on. And these can be estimated from these properties if we choose to do that. So let's start at bioavailability and look at what tools we have available. Certainly, most of what we know, we know from pharma, and we can broadly talk about these 4 types of absorption organs, for lack of a better term. And some classifications couple eye and skin together. I've chosen to show them separately. But certainly, understanding the physiology of each of these organs does help us understand what can prevent a chemical from becoming bioavailable through one of these potential pathways. So we can look behind that physiology and look at what the properties, the physical-chemical properties that we can calculate, are associated with lack or bioavailability. So for example, for bioavailability through the GI tract for absorption, we need molecular size less than about 500 Daltons. We need a log P of 0 to 5, and we prefer non-ionized molecules that can cross the membranes. For skin bioavailability, we have a different size range, about 400 Daltons, log POW, octanol water partition, about 0 to 6, and certain ionization state are preferred, such as polar ionized water-soluble molecules. So I can go around and tell you about the rest, but I think the point here is that certainly there are some differences of in between the ranges of values that favor absorption or disfavor absorption from the 4 organs. So if we know that a chemical is likely to be present and exposure is likely to happen through one of these pathways, we have pretty well-defined guidelines for what the properties would have to be in order for it to not be able to cross these biological membranes. And of course, Lipinski from Pfizer certainly came up with the so-called rules of 5, the Lipinski rules of drug likeness, based on very similar principles. And so he observed that of the 800 or so drugs that Pfizer had put into production, a lot of them met certain very simple criteria for physiochemical properties. We know that those are associated with good absorption through the GI tract for the most part, and there's been a lot of new papers coming out that deal with how these properties are evolving. But the key take-home message here is that these are essentially design guidelines for making a good drug. So our goal really is coming up with similar design guidelines for making a compound that's not biologically available and does not hit biological targets. So the next number 2 stage of the previous diagram was distribution. So how do we think about properties of a chemical that make it not be distributed quickly or easily throughout the body? One of the key ways we can think about it is using the volume of distribution, which is just a theoretical concept. We're talking about how much of the chemical is in the body versus how much is in the blood. But the key take-home message here is that water solubility and binding to certain blood proteins are some of the key features that can distinguish what a chemical that's distributed well would be from one that isn't. And so this is something that we can use quite readily, and a lot of toxicokinetic modeling is being done to this effect to model volume of distribution. There are some special membranes that we have to consider, such as the blood-brain barrier, where we have different characteristics of that membrane that exclude a greater number of compounds. And we have good examples, for example, the one I've shown in this scheme here, where methylmercury is able to mimic essentially a cysteine molecule and enter the blood-brain barrier. So there are exceptions to some of these guidelines, but again, they're a basic 80-20 rule for what can reduce distribution. So we move on to bioactivation. And again, a lot of chemicals that we're exposed to have to be metabolized to something more toxic in order to exert their ultimate effect. My perhaps favorite example is benzene, which has to be metabolized and oxidized up in order to interact with DNA and other biochemical targets. So how do we think about bioactivation? Now, this is progressively getting more and more challenging as we're dealing with more biological complexity. But just to give you a small example, P450 is responsible for about 90% of phase 1 metabolism, and it's one of the key enzymes that we're concerned with. Certainly not the only one, and there's also phase 2 metabolism, but as an example, we can break down the way in which P450 takes a molecule, metabolizes it into something more toxic or less toxic, let's say, and then releases it. And so we're modeling a binding pocket here where we have different parameters that control the desolvation of the chemical, different parameters that control the binding, parameters that control the turnover, and ones that control the release. So you can see the complexity is building up quite quickly. And I've listed some of the parameters that are associated with each of these steps. We're talking about log P, hydrogen bonding, certainly pi-pi stacking interactions, pi surface area. So I can't give you a simple guideline for what this would be, but I can tell you that a lot of good computational work is being done. And for example, we're looking at, and other groups are, at energies that can be calculated quite readily of the radical stability of various molecules. And this is quite, again, a good predictor for what can be metabolized readily by P450. And again, we can come up with surrogates that can tell us what a stable radical would look like. And so this is sort of the qualitative SAR approach, if I can put it that way, of thinking about what it would take for a chemical not to be metabolized. So again, just as a summary, some of the key work in this area that's been going on for many years in human P450 metabolism has shown that frontier orbital energies are a key parameter that we can calculate quite readily from computational tools. Steric hindrance, molecular shape, and redox potential. And the one I mentioned here is radical stability. And so we do have the tools. This is in detail described in publications that I'll give you at the end. So I'm not giving you the gruesome details. I just want to highlight what are the key parameters that can be informative to us for bioactivation. But finally, we get to the stage 4, which is the most challenging, and that is looking at actual interactions of a chemical or its metabolite with biological molecules that are present in cells. And here we're talking about proteins and their side chains and residues, RNA and DNA, various enzymes, for example, causing suicide inhibition. We're talking about receptors. And so the number of molecules that we're referring to here is huge. But there are some similarities among them. And so I'll show you our example of trying to come up with some design guidelines for one class of molecules that are known to have interactions with some of these biomolecules. So we were looking at olefins and their metabolites, which are epoxides. And the reason is that, of course, olefins are found industrially in a number of important products. And we had some good data to start from. But the question we wanted to ask was, is it possible to come up with a rule of thumb for what an olefin that's not mutagenic would look like? Simple olefin, not part of a very large molecule. We just wanted to test the hypothesis that it may be possible to come up with such a design guideline. And so I was really inspired by a paper from the 1980s from Robert Jones that, back in the day when computational chemistry was just getting started, he was able to show that if we look at the bond strength of the carbon-oxygen bond of the corresponding epoxide to an olefin, he was able to observe some relationship between mutagenicity and the corresponding carcinogenicity of basic epoxides and that bond strength. So that was really inspiration for us to start thinking that maybe we can come up with more sophisticated computational methods to predict what might be mutagenic. So we looked at reaction coordinate diagrams. So we looked at, first of all, what is the key molecular event that causes mutagenicity. And of course, if you're talking about an olefin going to an epoxide, it's that reactive epoxide that's opened by biomolecules such as DNA and then can cause mutations. And that's well documented in literature with a couple of exceptions to this general mechanisms. So the key event that we want to model is an SN2 opening of that epoxide. So that's how we're simplifying the question to something that can be computationally handled. And so now we're looking at a reaction coordinate diagram that has both a kinetic component and a thermodynamic component. And we're trying to understand the kinetic component because we feel that's the more sensitive one to look at. And to do that, we basically are looking at 14 different epoxides with various halogen and alkyl substituents, and we're calculating what that energy would be. So we're using various methods, Hartree-Fock and MP2. MP2 is a more sophisticated and computationally intensive method for doing this calculation. And here I've just compared the results of these two methods, showing that they're actually comparable for what we're looking at, with the difference being that molecules that have an alkyl group are much better handled by MP2. So what does the result tell us? Well, we have one molecule in there which doesn't fit our trend, and I'll tell you straight up, it's ethylene going to the unsubstituted epoxide. But for the rest, what we observe is that an activation energy of about 5 kcals per mole is what divides the mutagenic molecules from the non-mutagenic ones. And so that essentially tells you that if you calculate an energy of greater than 5 kcals per mole, and I should tell you that this calculation takes about an hour or an hour and a half on a computer max, then based on the data we have, it looks like that's a good predictive tool for an epoxide that's not mutagenic, and vice versa. But not everybody can do that in their lab, and certainly we would like to have surrogate parameters that are able to correlate to this kind of kinetic barrier. And so we've examined several, and I can tell you that the best ones and the ones that are easy on a computational level to calculate are the difference between the HOMO and the LUMO energy levels, and that's really talking about the electrons that are doing the business end of a molecule, the last occupied and the first unoccupied orbital level, and so that's the DE that you see here. So what you're seeing here is a separation between the molecules that were non-mutagenic and the molecules that were mutagenic. We can also look at the charge on the carbon, on the unsubstituted carbon, and see a difference, and of course we have one outlier. And again, we can look at both the olefins and the epoxides and their dipole moments. And on the olefin, we see a significant difference between a dipole of the compounds that were non-mutagenic versus ones that are. So the point of this study is that we can come up with design guidelines for one class of molecules that corresponds to their mutagenicity. And again, this can be repeated, I think, for different classes of compounds and for different endpoints, but it has to be done on a case-by-case basis. So this is my vision of where this field is going, and I'm glad I just saw Ruben walked into the room. I was a computational colleague, and actually I'm not a computational person myself, but I try to meddle into this a little bit, so he can speak to much better extent than I can about this. But we started a long time ago at a time when computational chemistry could only address small molecules. Toxicology, a long time ago, was mainly concerned with whole animal models. And I think over the years, what we've seen is that both of those fields have started to converge, and computational chemistry now is able to tackle problems at the enzyme level and even organelle levels. And toxicology has given us an understanding at the molecular level of the interactions. So this is where I think this field can really be advanced for coming up with design guidelines for specific classes of molecules that are known to have specific interactions. But that's not all I'm going to tell you today. There's something else that we've been working on that I think is quite hopeful and exciting for areas where we don't know all the mechanisms. So we're talking about combining these guidelines for steps 1 through 4 into one set of guidelines that you can look at one time. So the way that we've thought about doing this is through what we're calling a hand-in-hand mechanistic and statistical analysis. And before I go further, I'll tell you that it's not a QSAR approach. QSAR aims to model a toxicity parameter and give you a predictive value. We're not doing that. We're simply trying to come up with guidelines and say, look, what distinguishes the highly toxic from the most likely to be non-toxic compounds is this parameter. So just to be very clear, it's not predictive. And what we were really interested in looking at first was aquatic toxicity, because that's where the data is, and there's a lot more great data coming out on this. And so in trying to tackle first acute aquatic toxicity, we had some great data from both the US EPA and the Japanese Ministry of Environment on diverse species. We're starting from a microalgae down to a fathead minnow and to a daphnia, which is a crustacean. So I think most would agree diverse mechanisms within the aquatic range of species. We categorize this data based on the threshold level, the LC50 or EC50 value, based on EPA categories from red to green. We've looked at the mechanisms by which some of these chemicals exert toxicity. For data where that is available, of course, there's a beautiful data set from the EPA on fathead minnow toxicity. And all I can show you from this graph is that there is no one mechanism which is very different in toxicity from the others. It's not enough to know about the mechanism to understand which set of chemicals might be more toxic. But we explored a variety of different properties, and so I'll give you just a summary of that analysis, which is the following. We know that log P has to be important. The octanol water partition is key to bioavailability. It's key to narcosis. But what else can we use? What other property or parameter to come up with the reactivity parameter? So we've looked at various different ones. LUMO energy is here, and delta E, the difference between HOMO and LUMO, is here. So here are 3 different species. You'll see that we see the same trend between the 4 toxicity groups for log P. And for LUMO, you see that we don't see the same trend. So we have a nice trend for fathead minnow, not really much of a trend for the other 2 species. But if we looked at delta E, the difference between those 2, then this is actually very little data in this last group, so this is hard to see. But we see the same positive trend with Daphnia magna, we see the same positive trend with fathead minnow. So that was a potentially useful property that we could use. So if we look at this in chemical space, let's look at the combination of these 2 properties together. So this is fathead minnow, Japanese medaka, and Daphnia magna. What you see here is that all the green dots are the group 4 molecules which are least toxic. And the red, actually, I apologize, I've sliced these halfway. So green are groups 3 and 4, and red are groups 1 and 2. So toxic versus non-toxic, red is toxic, green is non-toxic by EPA cutoffs. All the green molecules are in one quadrant. And so, there are very few green molecules here. You can't really say much. But again, the same for the Daphnia. The same quadrant that's cut off by the same property values for delta E and log P. Now, we actually came up with these property values by recursive modeling. I didn't just draw a line here. But if we combine all of the data together, then we see the same here, where all of the green dots are in the same space. And yes, there are many red dots in that space. Let me be perfectly honest. But what this tells me as a chemist is that if I'm designing a new chemical, and I want to have it, with a log P of 6 and a delta E of 6, my chance of making a safe molecule is probably very, very low, because none has been made so far that is in that space and that is safe. We can look at the same thing for the algae. Different mechanisms of toxicity, different bioavailability. The same green quadrant that you see there contains very nicely all of the compounds that are known to be safe, acutely safe, to this algae. And so just for comparison, we've looked at some of our traditional bad actors that we all know about, BPA, phthalates, atrazine, and so on. And I've given you their characteristics for log P and delta E. And I think you can convince yourselves that they definitely fall into the space that we would call toxic, which is all of these 3 other quadrants. So why is that? Well, very simply put, log P, I think we understand and we can agree that has to do with bioavailability and bioaccumulation. But if we look at frontier orbital energies, it's not such a simple case to make. Of course, most of the interactions I told you about that deal with either DNA modification or binding to proteins, but not receptor binding, I should say, do deal with an SN1 or SN2 process where you have an electrophile-nucleophile interaction. And these orbital energies here deal with exactly that. Your HOMO energy controls your nucleophilicity. Your LUMO controls your electrophilicity. And a molecule is usually either one or the other. It's usually not both. And so if you have a low-lying LUMO or a high-lying HOMO, you're going to be more reactive, essentially. And that's where I think the delta E parameter comes from, in that if you have a larger spread between those 2, you're less likely to be either an electrophile or a nucleophile. So if anybody has any other thoughts on that, I'm all ears to hear. That's at least my theory of what that connection is. So what does this actually mean? Well, I've summarized our results here, and we've grouped what the compounds that fall in this space look like, what the compounds that fall in the opposite quadrant look like, and what all the compounds in our data set look like. And let me just show you that this is broken down by the 3 different species. That basically, the compounds that fall in this space are much less likely to be toxic. And one of the measures you can choose to look at is this is the mean and median LC50 values. So, for example, if you compare 1172 to 14.8, that's one comparison of how much we're getting a reduction in toxicity by using these design guidelines. And you could visually see that all of the safe compounds were in that one space. So, fine, for acute, maybe that's good and dandy and easy, but can you translate that to chronic aquatic toxicity? That's the hard question. Well, we have, and I don't have time to go into all the details, but this is a very similar graph to the one I showed you. We're using different data for chronic assays that we have about 1,000 data points, 1,000 compounds. We had about 1,000 for the acute. And what I'm showing is that most of the green compounds are in the same space. And these are, again, by EPA categories of chronic aquatic toxicity. But we have some outliers, and we're very interested in understanding why they're outliers. So these are your phthalates. And your phthalates are green. And I'm sure we have probably going to have some thoughts about whether they should be green or not. We also have some thoughts about whether that toxicity was measured accurately. Phthalates are known to have certain interactions in aquatic systems. And so we have several theories why they're all the way out here, but they are claimed to be green. And also we have some molecules that are certainly red. Only a couple, actually, that are red that are in our so-called safe space. And we have theories on how they hydrolyze it and why pH and pK factors that contribute to them being toxic despite these properties. So this is, again, an easy way to understand how much of an improvement we've made by looking at the compounds in that safe space. So if you look at the median log P, the median NOEC value of all the data, it's 0.48. If you look at the compounds in the safe space, it's 4.2 versus the compounds in this opposite quadrant, 0.084. So again, we are seeing a significant improvement in our ability to pick out a safe compound from the quadrant that I've highlighted here. So in summary, I think you can see that we have some tools for being able to understand what makes a compound toxic to aquatic systems. We see 70 to 80% of our safe compounds appearing in one quadrant, and that's a big revelation to me as a chemist if I'm trying to make a new molecule. And again, the cutoff values that we're using, that we're suggesting, are log P less than 2 and delta E value, which is again predicted by semi-empirical methods, which are quite fast and easy to use, greater than 9 electron volts. We've applied similar strategies that I won't tell you about here to avian toxicity, and we've done some work on various groups of chemicals, including pesticides, herbicides, and fungicides, and try to understand what are the factors that control avian toxicity. They are different. They're vastly different from the ones for aquatic, but they can certainly be rationalized as well. And I'm just showing some eye charts here of how we've broken down various chemical groups within avian toxicity data set, and to understand what are the most likely set of parameters that is likely to give us a non-toxic chemical, and its globularity, water solubility, a number of rotatable bonds. I won't go through this one, but one of our early pieces of work was trying again to use this methodology to distinguish chemicals in the EPA toxic registry inventory from a randomly selected chemical. And by randomly selected, we're talking about looking at about 11 million compounds in the zinc database, which pharma uses to data mine. And so we were able to show by, I would say, very simple statistics that we can distinguish what a chemical that's belonging to the toxic registry inventory would be from a random chemical by 2 properties. And these are, again, the octanol gas partition coefficient and polarizability. And so what this tells you is that if you start with 2 groups of chemicals, one belonging to the toxic registry inventory and one random, then those that have a lower octanol gas partition coefficient of about 12, and also ones that have a lower, greater polarizability than about 17, are much, much more likely to be members of that group. And in fact, 471 of the 571 TRI chemicals are scooped up by this first cutoff. So there's a lot of issues with this as well, and not all chemicals on the TRI list are necessarily equally toxic. But I think there's a lot to be gained from looking at the data that we've collected from a new perspective and trying to ask the question, what distinguishes the toxic chemicals or the ones that hit a certain endpoint from ones that do not, rather than how do we model it as a QSAR. So I hope what I've showed you here is that there are certainly many challenges that we face going forward, but we have some hope and we have some tools that we can use today. And I think with development of new computational tools and with new toxicity data that we're going to incorporate into our models, we're going to be in an even better place to address this tomorrow. But with that, I just hope that we can walk away with the fact that this is something that we can do starting today, and hopefully have more to report back to you in the next couple of weeks or years. So I'll just leave you with my vision of what our holy grail is in this field. And right now we're talking about using properties that we can predict. I think ultimately we as chemists would like to think about what we can measure. And that's one way that I'm heading with my own research, is hoping to find ways that we can use measured NMR data, measured mass spec data, and characterization data from materials, and try to understand what the relationships between those measured parameters and toxicity are. And so, it leaves a lot to be desired, but I think if we can do that, then we can give chemists tools that they can readily use to be able to do this. So with that, I'd just like to thank you for the invitation and for the opportunity to share the floor with such a distinguished group of speakers, and I'm happy to take questions.
Thank you. We're running late, so just one quick question, George, please. How can I not give George Daston?
George Daston, Procter & Gamble. See, I did it. So I think this is the right thing to do to develop these principles. One concern that I have is that you haven't talked at all about weak interactions. And so, obviously, I think we can get our hands around covalent reactivity. It's the weak interactions, the receptor binding, where we're going to find our most potent toxicants. And I'm wondering if it's worth taking a step back from these highly quantitative measures to put in perhaps more qualitative measures or qualitative design principles, which is probably the best that we can do, except for those very few receptors where we have the crystallography data.
Yeah, thank you for that question. I couldn't agree with you more that there's a lot of weak interactions that are key to many toxicity mechanisms. There's a lot of great computational work being done currently to model some of these systems, and I think we can use that data in a similar way that we can use toxicity data to try to understand what are the parameters that are most associated with strong binding. So my example would be for endocrine disruption. A couple of parameters have been published that are most highly associated with estrogen binding. And so I think we can apply the same idea to that, but certainly a very important one to address. I only had time for one example, but thank you for that.
Thank you.
Okay, thanks.
Our next speaker is a colleague of mine from UNC Chapel Hill. Alex Stropsha is a Associate Dean for Research in School of Pharmacy and has been working on computational toxicology methods and QSAR for his entire career.
Thank you. Where's the pointer? Here. I appreciate the opportunity to see Ivan outside of UNC Chapel Hill. So let me jump right in and give you the roadmap for this talk. I guess my main point, and that's what I was asked to present, is that I'll initially talk briefly about an elephant in the room filled with biological experiments, and that elephant is a chemical structure represented explicitly, and that's the core of cheminformatics. To recognize that this knowledge can be explicitly encoded and used in analyzing data and talking about in vitro to in vivo extrapolation. And so I'll have a few introductory notes on how cheminformatics fits into what we've discussed so far. And in that regard, I'll talk about predictors of endpoint toxicities, historic predictors, relying on the information that we've had for years of structure-activity relationships and building QSR models, but also emphasize that the new data streams that come from omics experiments supplement, or they mutually supplement, chemical data streams and biological data streams do not compete, but actually complement each other. And I'll talk briefly about building what we call hybrid models that utilize both explicit chemical information and biological information. And in case Ivan wouldn't allow me to finish because of time constraints, I'll give the conclusions right now, and that is that we have diverse data streams that require novel data analytical workflows, and I hope to introduce at least some. I'll emphasize the utility of models, which is that they're used to prioritize or design the desired chemicals. And what I firmly believe in, that models that deal with safety, specifically toxicity, safety, must be made public with all the proper data. And we're doing our share of this by building a web portal called ChemBench as a freely available in silico toxicity profiler. So I'll just show you what it is. This is a busy slide, but it kind of outlines our approach to data modeling. And by I guess, remembering some of the analogies from this morning, I'd like to think of computational chemists or computational modelers as those who stand on the shoulders of giants. And those giants are people who generate data. Without data, which is critical to what we do, we wouldn't be enabled to build models, analyze this data, and hopefully help the giants do less with less. I guess generate less data, but of higher utility. And by the way, I was thinking, sitting in the back of the room, that all of us are giants. Because with this room configuration, you have to be standing on our shoulders to see the slides. So I'll have to. I think you're going to see half of my slides, understood as half of each slide. Okay. So let me walk you through this. We start with chemical substances. And thanks to efforts such as ToxCast and other PubChem and other public databases, there is an ocean of data. And this ocean comes into workflow where we analyze data, both structural and biological. And something that has been overlooked frequently is that the data cannot and should not be used directly as deposited. All these public databases are repositories with sometimes insufficient effort spent on curating and harmonizing them. And so it's important to our workflow, and I will illustrate with at least one example, that the data collections before they become data sets must be procured, and both with respect to biological and chemical data. And the data streams create two formally independent ones, which is chemical descriptor-based, which is cheminformatics, and biological short-term assays, which we regard as biological descriptors or biological features of chemicals that we use in building our models. And so that goes into machinery which processes this data and builds statistical models with a lot of validation protocols incorporated into the workflow. And that leads to validated models of in vivo endpoint toxicities that we port into our ChemBench. So for those of you who only see the title of the slide, that's a story on the rest of the slide. Okay, where does it lead, and what is the utility of models? The input is biological activities or properties of chemical descriptors. The outcome is decision support. And one aspect that I'd like to emphasize is that models that we build are statistical in nature, and they are produced to make statistically significant predictions about collections of chemicals. They are based on collections of chemicals, and they are best used and utilized to prioritize subsets of external libraries or designed compounds for specific confirmatory biological experiments. And so it's not the same as to use these models to design a single compound, which is one of the desired properties of computational models. But yet they can be used this way as well. And so I'd like to emphasize again that models are used as virtual in silico profilers to prioritize compounds for subsequent biological experiments. Something that is in the core of our methodologies is, as I mentioned, explicit representation of compound structure by large collections of chemical descriptors. And I'm just using a few of those. Memorize the slide because I will illustrate later, few minutes down the road, that exactly same type of input table can be built with biological data resulting from short-term assays. But that's what goes into the model, which is a slightly different representation. Chemical structures are translated into descriptors. Statistical data mining, statistical models are used to to relate values of chemical descriptors and values of measured biological properties, such as endpoint toxicities, which can be real values, binary or categorical parameters. And so let me talk about the first aspect of careful approach to looking at the data before any models are built. And I've mentioned this analogy with standing on the shoulders. And so we are not only standing on the shoulders of giants, we're at their mercy. And what I will illustrate is that prediction performance of statistical models in general strongly depends on the quality of the input data, both structures and activities, both endpoint data and accurate representation of chemical structures. And both of this data may be inaccurate, and we have to be careful to curate the data sets. And that influences what we publish. And I would claim that there are models that have never been published because they were bad for their own reason. And there is a large variety of models that have been published for their own reason either. And so this is something to we have to pay attention to. And so here is an example of our study of Ames data set. And if you just visually inspect some structures in the data set, you wouldn't see anything wrong with the structures per se. But if you submit these chemicals to one of the most popular and robust software to generate molecular descriptors, which is Dragon from Milan Chemometrics Group, it will tell you that every single structure cannot be processed because the structure would have either errors or there would be duplicates, which you should not use in model development, or there would be types of molecules that standard cheminformatics software cannot process, such as organometallics or mixtures. And the point is that the structures cannot be used. Or in some cases, wrong descriptors would be generated, and that will lead to wrong models. And this is a summary of what could go wrong. We could have erroneous or wrong structures with the wrong chemistry. We could have presence of misprints or wrong names. So in the list of biological activities, that all will be kind of low numbers. So it would have a substantial outlier that's likely to be a typo, and we see a lot of this. We would see duplicates that will artificially increase the predictive performance of models if one of the duplicates will end up in an external evaluation set. We'll have mixtures that require special attention, and typically protocols would randomly choose one of the structures as the true representation of the mixture, etc. So we've certainly will not generate rigorous models based on this. And mindful of that, we published a paper last year with the title Trust But Verify, which emphasizes our approach to the data. It's also a famous quote from Ronald Reagan that he borrowed from the founder of Russian KGB, Felix Dzerzhinsky, which is why I'm familiar with that painfully well. But anyway, all these protocols have been integrated using publicly available for academic lab software, and it's been made part of standardization that's part of ChemBench data processing. Give you one example, nitroaromatics. Of course, important compounds. Strangely enough, these compounds are subject of green chemistry. For some reason, we don't worry about eco after explosives go off, but we do worry before they go off. And so if you look at these compounds, these are different ways. It's a very, very subtle aspect. How do we represent a nitro group in nitroaromatics? Turns out that depending on how we harmonize the representation, the outcome of descriptor calculation will be different. And if we don't harmonize, as simple as that, the models, the external power of models built for different cases would increase from 0.45, which is not very significant, to 0.9 in one case, or from 0 to 0.5 in another case, only because we would harmonize the representation of chemical structure, as subtle as that. And so this one example, we have tons in that publication. Data curation impacts the true accuracy. It could be either up or down, but without data curation, the accuracy of the model would be artificial. Okay? And again, if you have duplicates, you're guaranteed to have artificially high results, for instance. So it could go both ways. Let me give you a few examples of purely structural workflow, and that's historical data collections that are relevant to various endpoint toxicity. So we use chemical substances, procure them, calculate chemical descriptors, build models. Aquatic toxicity, one of the data sets, and I'm using kind of random examples from a bunch of models we've developed over the years just to illustrate how we go about data processing. So in this case, All data came from the same lab, collected over multiple years. Even though it came from the same lab, we have to correct more than 100 errors, including chemical structures, names, Cas IDs, etc. We had almost 1,000 unique compounds. And publication that came after we put the collection together was used as an external set. One aspect of data processing is that I don't like to use the word belief because it's not a scientific term, by practice, let me put it this way, is what I call competitive collaboration, where we've put together a group of colleagues. Each group had independent approaches to QSR modeling. And we decided that we're going to collaborate to test different methods and develop the best model we could possibly come up with. And so we agreed, so 6 groups from 6 different countries. We agreed on simple metrics. More than half of you cannot see that, but this is training set accuracy, test set accuracy, and mean absolute error of prediction. And we evaluated all models looking at the external data set only. And so what you completely cannot see, but what was the result of this study, is that consensus model, when we integrated all models together, and someone talked today about the power of consensus, and this is one of the aspects of modeling and one of the aspects of our approach to data modeling, generation of the ensemble of model, and using the consensus as the predictor of the highest accuracy and the highest space coverage. We could predict the largest fraction of compounds. That gave us the highest accuracy for independent evaluation sets in this case. And the same type of study was recently reproduced with a greater team. So other groups had heard about it. So our first publication, we formed a collaboratory of 14 teams from different countries and worked on Ames mutagenicity, 33 models, 9 different types of descriptors, 17 modeling techniques, 6 different approaches to estimating the domain of applicability, which is how you restrict the predictive power of your models, not extending beyond certain groups of chemicals that have not been seen by models. So this is a very important aspect of any statistical modeling. And once again, what we saw is, and I'll comment on what we mean by external validation a bit later. What we saw was that the consensus model was the most robust, most stable to variations of individual models, and had the highest chemical space coverage. Once again, could be used to predict the largest number of chemicals. So last example, and that's very recent and not published, is that we've been working on developing models of estrogen receptor binding affinity. For the first time, from what I understand, we put together independent data sets to evaluate ER alpha and ER beta binding. And I use this slide to illustrate how we process the data. That is, that we use what we call external validation, not cross-validation, but external validation, when we take progressively 80% of the data set and do all the modeling and internal validation on 80% of the data, and collect the predictive power of models on the 20% of the data set that is never seen by the model. And we do this systematically, and we compare these evaluative numbers, not using the external predictive power in any way to tune the parameters of the model. And so what we saw here, and that's another opportunity for me to bring in some aspects of approaches to modeling, and I think is very important and critical to what we're trying to accomplish here, is that methods that we used belong to 2 general categories: single task learning, which is what STL stands for, and multitask learning, which is what MTL stands for. In this particular case, we saw that multitask learning approaches, which simultaneously learn from both alpha and beta examples, was superior to single task learning for the case of ER beta prediction. It's been comparisons made this morning between what we've done in pharmaceutical drug discovery and drug design and in green chemistry, and we're dealing with multifactorial optimization problem. We're trying to optimize. There is the direct analogy with computational approaches that are used to design drugs and at the same time have compounds drug-like and not having undesired admitox properties. And I think what we're trying to accomplish here, which is that compounds should remain active in terms of their primary application and yet lose all undesired properties. So it's clearly a case for applying advanced methods of multitask learning, and there are some examples, emerging examples, that these types of methods produce better models. Something else that may emerge from the analysis of descriptor profiles. So these are values of descriptors, characteristic values of descriptors for potent ligands versus ligands of low potency. And we see this is some representations we're playing around with, but we could see that the profiles are distinctly different for active versus inactive compounds, and we could use this knowledge to start thinking about inverse QSR, inverse design of molecules with the desired or undesired, with the desired ER affinity. And so this is just a snapshot of some approaches to some thinking, recent thinking in the lab as to what we could do. The utility of models, that's a collaboration with the ComTox Center. They talked about what's done in the center, and part of that work is selecting compounds for subsequent phases of trials. So we've used these models to prioritize potential estrogen receptor binders from a subset of TOX21 group, and this is how these models can be used for prioritization purposes. Let me finish off with what I've called hybrid approaches, or what we've been increasingly doing by combining the explicit knowledge of chemical structures, and I've just illustrated how this can be done to achieve models of high external predictive accuracy. And data, the omics data that emerge from a variety of short-term assays that collectively are regarded as toxicity signature. And the slide is it says courtesy of. That's a euphemism for mercilessly stolen from a colleague's presentation, which he shared with me. But that's from Ann Richards from ComTox Center. And so I've mentioned already that we emphasize that this data could be additionally enriched by the information that's already there, which is explicit representation of chemical structures and their properties calculated from the knowledge of the structure. It does not change the workflow. It changes how we present compounds. We have traditional representation of chemical descriptors. We add to this in vitro assays treated as biological features of chemicals. And I've mentioned that I will illustrate. I'll ask you to memorize one of the input tables for QSR modeling. So I'll show you another table coming from this specific study where we've looked at hybrid descriptor-based modeling of rat LD50 for substances for which we've had both cell viability assays, 13 cell viability assays from the NIH Center for Chemical Genomics that were tested. So chemicals were tested in this cell viability studies at 14 different test concentrations. And so when you look at this data, at the face value, this is a biological profile that can be used to predict in vivo profile. But we could transform this data into a familiar format where each cell line and each concentration would be equivalent to a name of a descriptor, such as molecular weight with chemicals, and the value, the dose, the response value in response to the dose would imply the value of the descriptor. So it's exactly the same format that we use in traditional QSR modeling. Something else, I've mentioned that it's important to curate biological data, and what we found by experimenting is that if we take raw data, which frequently look like this, non-ideal dose response curves, we could transform and streamline them, eliminate some noise, and that actually helps with the outcome of modeling, as I will again illustrate. I'll skip the protocol, but this is indeed a very important slide, and it emphasizes and illustrates the importance of using both chemical and biological information to improve the endpoint prediction. And so, as probably most of you know, when we developed QSR models, that's about similarity. Similar compounds are expected to behave similarly. But here is examples of how biological data help improve predictions made with, or mispredictions based on chemical structures alone. What's plotted here is similarity of compounds in their biological profiles. So you could compare biological profiles of compounds, and on x-axis, the positive direction is increased dissimilarity of biological profiles. Y-axis is chemical dissimilarity. And so we have cases of compounds that have nothing in common chemically, both are non-toxic or both toxic. And you cannot rationalize the outcome of the experiments based on chemical similarity. It's very low. However, both pairs of compounds tested similarly in biological experiments, and that's how biological data could enrich and inform additionally chemical arguments, purely chemical arguments. So back to statistics, what we find is that using chemical descriptors or hybrid descriptors without treatment, we generate models with correct classification rate, external rate of 74-76%. And using this processed biological data with noise reduction protocol implemented, we could grow the accuracy of models, not by a huge amount, but in statistically significant sense. And that again emphasizes how the use of both types of descriptors improves the utility of models. We also, and it's just very tempting to compare the results of this focused modeling exercise with the use of commercial, one of the popular commercial packages. And we've also found that when we used both types of models on the same external set, these models that are specifically built for this data set are superseded in their accuracy, the commercial predictor. And another aspect of this analysis is, which is very interesting, is interpreting models. So each dose is a descriptor, and we could analyze the results of modeling with respect to the importance of each dose in each cell line, statistical importance, as to the accuracy of toxicity prediction. And some interesting observations that come out of this is that you kind of expect that the higher the dose is, the greater is the contribution of the dose to toxicity. And generally this is true, but there have been multiple cases when lower dose would have greater statistical contribution to the endpoint toxicity. And so the results of modeling can be used to reprioritize both cell lines and specific concentrations in which compounds should be tested to achieve a high correlation with the endpoint toxicity. So my last two slides, as I mentioned, we develop models, validate them, publish them, and then we add them to this portal, which has a list of predictors. And in fact, all predictors I've mentioned today, Ames genotox, ER binding, hybrid models, aquatic toxicity, have been made publicly available. And so a user could upload chemical structures and biological data if they exist and execute the prediction. And finally, just final notes on the methodology. I emphasize the importance of data curation. I emphasize the importance of consensus or collaborative prediction using all acceptable models. And the outcome of modeling methodologically is that we're not making decisions, but we're building tools that support decisions in selecting future experimental screening sets. And in some cases, that's what we're moving into. We should be able to come up with a design, inverse QSR-based design of chemicals. I illustrated that the highest accuracy is achieved by models that employ both chemical and biological descriptors. So we emphasize this symbiotic relationship between short-term assays and chemistry. And as I illustrated, interpretation of significant chemical and biological descriptors helps inform the selection or design of novel safe chemicals. So with that, thank you for your attention. I thank funding, especially from EPA, who funded a lot of studies that I reported today, collaborators, students, postdocs, etc. And thank you for your attention.
We'll have time for questions, so let me just start, Alex. And so the Adelina's presentation was arguing for a few of the descriptors as potential design principles versus some of your modeling is using the more the merrier, of course, not the thousands. So how do you balance out some of the, I don't want to say opposing, but maybe complementary drives in this type of analysis that tries to combine chemistry to predict toxicity?
So thank you. It's a good question. I don't think there is any contradiction because I think what Adelina was talking about are a few physical chemical parameters that are easily understood and easily computed. Descriptors that we use kind of take into account finer aspects of chemical structure. Some of them cannot be directly interpreted. But for instance, log P is one of the parameters that's used. There have been competitions between groups like ours to predict log P from chemical structure. And so some of these parameters actually embed structural information. Something that I did not mention is that we rely on variable selection methodologies that emphasize a small collection of descriptors that are significant in the context of very specific biological properties. And those could be mapped onto specific structure. So I think we're talking about different level of chemical resolution, but I think it's fully compatible. These types of views and are fully compatible.
Okay. Please use the microphone.
Pete Myers, Environmental Health Sciences. It's really wonderful to see the advances in computational toxicology that you're describing here. Immensely powerful tools. It strikes me that one of their limits right now is that to do these calculations, to do these analyses, requires large data sets. And by and large, the large data sets are available only from standardized assays, classic assays. And yet, modern molecular genetics is way beyond those standardized assays. So that the approaches that you're taking now are based on old understandings in toxicology. How do we bring the two together so that, in fact, these computational tools are guided by modern molecular understanding?
So I think it's a very forward-looking question, I would say. And I have a slight sense that the man standing on my right will be able to answer that better than I do. But I'll try to start and he'll continue because it's my talk. So Ivan has been very interested in and pushing us to develop models that take genetic variability into account. And so we've been accumulating, or his lab has been accumulating data, not only this lab, that looks into genetic diversity, for instance, which comes from modern tools, and brings together the results of assays, the way we've done them done on different individuals, or cell lines from different individuals, such that chemical structure variability, genetic variability, and response variability, short-term response variability, are brought together in the same type of the same type of approach of synthesizing all this data to predict endpoint toxicity that is specific or genetically susceptible. I don't know if you want to add.
Well, this is Alex's talk, so let's talk about this in the break. Please use the microphone again.
Example.
Last question.
So such a great talk and really fascinating to see how far you can push that. So I just want to ask you about what seems like an apparent contradiction between. Sorry, I'm Ruben Abagian.
Let me introduce Ruben Abagian, computational biologist, #1 ligand docking specialist in the world, structure-based, etc.
I like to ask provocative questions. So I just want to ask you about this apparent contradiction between. In the beginning, you were saying that the chemical signatures would correlate with the predicted activity, with the biological profile. And then you said that the chemical signatures do not correlate with biological profile. So can you decipher it for us?
So they do and they don't. Okay? Okay, I'll qualify that. One of the approaches, and we're working on a paper now where we look in different ways of combining this information. And one of the approaches that we look at is to look at groups of compounds where there is a correlation versus groups when there is no correlation. And so I think what happens is that, purely statistically, you have a much greater space coverage, much larger number of descriptors. There is a meaning to both correlation and lack of correlation. Okay, and so I don't think I probably misspoke or was misunderstood by showing the slide as the way you interpret it, that there is a complete lack of correlation. There are groups of chemicals where there is a correlation, groups when there is none, and there is a reason behind both. So we are consolidating the information that we gather from these experiments in a way where both spaces assist each other in predicting the endpoint toxicity. But you would not expect a complete 100% compatibility for all sorts of reasons. Metabolism would be one.
Okay, thanks.
Thank you. So our final speaker in this session is Ed Carney from Dow Chemical Company. Ed is just another example of how reproductive and developmental toxicologists are now all computational toxicologists, because I know some other people who spoke in this session who were trained in reproductive developmental toxicology, but now are pushing the limits in predictive toxicology as well. Ed's task is much more difficult than others. He was charged with criticizing everything we've heard already about, and hopefully he'll have some constructive suggestions to that. So, Ed, looking forward to it.
Well, great. It is great to be here. Great honor, and very excited to just be part of all this too. I would say that description of the task is a little bit more daunting. So I'm not in any position as an expert to do much criticizing here, but just kind of give you more of a kind of a ground view especially from the point of the view of the chemical industry. So really my task was to, again, talk about levels of confidence. And the way I thought I'd approach this as more of my career in developmental toxicology and sort of in the trenches of regulatory testing and mode of action work is to start from the point of view of where we are now with what I would call traditional methods. I hope no one's insulted by that term, traditional methods. But because really in the end, we don't want to sacrifice the level of confidence we have now. We want to at least be there or higher. So I thought, well, let's step back and look at what we have now and say, well, what is it about traditional toxicology that gives us the level of confidence we have now? Because maybe we can learn from that. And then again, try and talk about this in a context of more of a practitioner's context from the chemical industry in particular, and then just have one slide at the end talking about how we can kind of move forward and develop increasing confidence in the new approaches. So this is really I thought this slide kind of pictured in one little slide where we are at today, and that we have again the traditional tox over here, and I put high confidence, and I guess you could debate that, but it is what it is. And we want to be over here in this tox testing in the 21st century, and ultimately we want to feel confident in that too. And you see here we have the brave souls, a lot of them in this room, that are willing to go and make that journey. But not everyone feels that way about it. There's any range from thrilled or nervous, scared, terrified. Those who are just a few years away from retirement are probably more in this category. They're just not going here at all. But the question is, how do we get across this bridge? So I'm going to start with a traditional toxicology approach. This is one that I would probably about 5 or 6 years ago from my lab, a compound called Dynacap. Maybe some of you have heard of it. Commercial name is Carathane. So I bring it out as an example of what I would call rather high confidence in using traditional approaches and to find a substitute for Dynacap. So this is a compound that I think Dow inherited from a company we probably bought. I think is how it started. But it's a fungicide used particularly in Europe in the vineyards, particularly in Italy. It's been known for many years to be teratogenic in rodents in the standard regulatory testing. So it caused cleft palate, torticollis, or it was a head tilt, circling behavior. And the no effect level, the lowest one for DevTox was 4 milligrams per kilogram per day in mice. And so if anyone's taken physiology or even had kids, you know that when you have an inner ear infection, it can cause this sort of imbalance. It causes the head tilt or the circling behavior, that kind of proprioception effect. And so it was tracked down. Actually, some very nice work done in the 80s, done by EPA, John Rogers' group, showed that it was caused by otaconial agenesis. So if you haven't heard of an otaconia before, this is a depiction of the inner ear. And there's two parts in particular, the utricle and the saccule. And they have these literally calcium carbonate crystals that sit on these membranes. And they sort of they're called affectionately as ear dust. So they put sort of a weight on these receptors, and that enables you to sense linear acceleration and proprioception. So, whoops, there we go. So really the goal, though, was, back in this time frame a few years ago, that we had to phase out Dynacap. It was under pressure in Europe, and the Italian wine growers were going crazy because they didn't have a substitute. So the search was on to find a less hazardous substitute. And so just so you can, I don't know if you can really see that, but what you should be seeing there is the normal, so the little sort of lit up, sort of like an X-ray, where you see the white otaconia there, and there's none in the Dynacap treated one. So kind of an interesting, not a typical effect you see every day. So what do we do in the search for these substitutes, though? Where do we start? Well, one of the things that has been known for a long time, Dynacap is not really just one molecule. It's composed of 6 different isomers. And again, pointing to the work done by John Rogers at EPA, we knew that 2 of these 6 isomers were not teratogenic. So that's a major start there. The other big thing, we talked a lot all day today about sort of the convergence of the chemistry side with the toxicology side. Well, this is a good example of that. The chemists found a way that had been elusive for many years, but found a way to scale up the production, isolate these isomers, and also produce them in larger scale. And without that, none of this could have happened. So with that, we were able to develop a mouse teratogenesis screen, so an in vivo test, so a short-term test, to test the isomers individually. And to make a long story short, we found one of the isomers, the 6-methylheptyl isomer, was selected as the lead candidate for substitution. Then we then proceeded and tested it in mice with full developmental toxicity study, did not see any effects. In fact, the cool thing was, we went from 4 milligrams per kilogram here to the Dynacap to this isolated isomer is 500 milligrams as our no effect level. Went on to test it in rats and rabbits. And the amazing thing was that so we not only removed the toxicity, but it also still had the same efficacy as in the field. So that made everyone happy, and it's a good day when you can make your Italian wine growers happy. It's a kind of a real win-win for all of us. But we were able to commercialize this product as a non-teratogenic replacement. So why did that work, though? That's really my point. It's not trivial, just a trivial example here. How does it relate to what we're talking about today? So one reason why it worked is that we had a nice assay for the specific adverse effects, certainly cleft palate and the otoconal agenesis. Decades of experience with the tests, including historical control data. A lot of people talk today about just the general value of a whole animal model, so I don't need to repeat that. But certainly the integrated biology, metabolism, pharmacokinetics, we had that. We didn't really quite understand it, a bit of a black box, but we knew it was there. And then we were able to confirm the safety in 3 different species. And then also we had the species conservation to humans on a basic level anyway. These mechanisms of development are highly conserved. And then the big thing I would say, really though, is that we had those prior studies from the EPA group that pointed us to the individual isomers. And I think this is a real, to me, from a practical level, a real eye-opener, because we search for substitutes and sometimes we talk about moving to a totally different class of compounds, but often the solution is right there in the same class of compounds with some minor tweaks. Okay, but so just to sort of pose a challenging question. Could we have done this using the tool, all of these nifty tools that we're talking about today? Could we have done it? And I would say it's probably be pretty difficult. What assay would you use? Do we have an in vitro? Anyone have an in vitro assay for otoconal development? I haven't seen one yet. The endpoint is very complex. We don't really understand it, but we know that this calcium carbonate crystal formation is clearly involved in it. But how do you duplicate something like that in an in vitro assay? And really, even though we know how these crystals form, we didn't really know how what the mode of action of Dynacap was, and we still don't really, nor do we know about metabolism. So, did I come here to sort of put a big cloud over the whole conference? Of course not. But where do we go from here? So, what's the case for using these rapid toxicity tests? Why not just use the old methods? Well, clearly, I think we all know the answer that we might have whatever level of confidence you want to ascribe to today's toxicity testing methods, but that's not going to move us forward in the field for a variety of reasons. Why? Obviously, it's slow, it's expensive. Really, it's set up for one chemical at a time. And I think the most important thing is it doesn't really get us anywhere in terms of mechanistic understanding. It's all descriptive, all kind of empirical. And that's not what we require for green chemistry. We need that speed, low cost per compound. I would add, since I was asked to give a chemical industry perspective, the speed thing is, just even the marketplace drivers for substitution are huge, and speed is really essential in that space. We have to evaluate groups of compounds. We're going to look at lots of candidates. And again, reinforcing this idea that we need to build our knowledge basis. And that is to me, what's so appealing about the new approach. And so how do we apply this in the chemical industry? Well, I sort of think of it as in 2 subcategories. The one is like the Dynacap situation, where we start with an issue with a compound, and we're looking for a replacement for it, a less hazardous replacement for it. The other one is just starting more from scratch, a little more proactive. How do we just get interjected into early stage R&D with the chemists? Again, like others have talked about before me, about how do you bring data to the table that the chemists can really use. So we have, it's been talked about a lot today about how this is sort of coming out of the pharmaceutical industry, but it's not exactly like pharma either. I would say, for one thing, I guess it's fairly obvious to this crowd, is that when we're designing chemicals that are used in the chemical industry, we're not looking at a human target or specific receptor. We're looking at things like how do I have flame retardant properties in an insulation board, or a conductor in a solar shingle, or for the golfers out there, how do we put certain chemicals and they make your golf ball fly straighter. I'm not a golfer, but those sort of everyday uses, but they're not human targets. Also, we don't usually, even if we have existing data on descriptive data from the regulatory tests, we rarely know the mechanism of action. And also, we just have such a large and diverse set of chemistries to deal with. So, like I said, I'm not here to put any gloom on this at all. In fact, it's just kind of to show the level of interest in the chemical industry. Certainly our company is a lot like Mark described in terms of some of the programs we have on sustainable chemistry in a larger sense. Also, what I'll drill into for the rest of this talk is that we've launched a program to bring in some of these newer tools in-house. And I think that that is a key missing link. I think even Pete Myers just said, how do you get this new data? Well, we've recognized the need that we have to generate this data, that it's not going to be out there. So it's intended to support green chemistry. I won't go any further with this, but a big application of these tools is also for Europe and the REACH program. You've heard of read across. There's huge, I think, a huge potential application there. And then just to be part of this general movement toward 21st century toxicology, we totally realize the world is changing and want to be a contributor to that. So that our program, which is, I'll admit is very brand new, just started last year, but we have a lot of the elements already, or we're designing them as we speak. These elements that were all talked about in the last 5 or 6 talks. So we have in vitro assays, biochemical assays. We have cheminformatics. That is, in kind of very early stages, but clearly that's in our vision. Model organisms. We're starting up the zebrafish model, for example. We've had a lot of other assays. Like my area is whole embryo culture. We've had that for like 15 years, and we're doing some work in the area of toxicity pathways. So we're still in early stages, but I think even this last year has been so exciting and just mind expanding, and think about the challenges that face us and how we're going to really make this work. And so one assay, or where I want to start now, is take you from the most sort of dismal situation to one areas where we have more confidence. And just as an example of that is the endpoint of skin sensitization. And so we have a number of in vitro assays. One in particular we're working on now is the antioxidant response element assay. So it's a reporter gene assay that was developed by Andreas Nasch at Givaudan. And sensitization clearly is a very important endpoint for a lot of areas, but in particular when we sell into cosmetics and consumer products, that kind of area. It's a real driver there. So one of the things that I think sort of philosophically, as we sort of build levels of confidence, is start to think about endpoints or adverse effects where we either know that either there's relatively few steps. There's not a single endpoint that you could say is not complex, but there's a relative complexity. And I think of sensitization as one where there's a few key endpoints that really differentiate sensitizers from non-sensitizers. I mean, I can contrast that to, say, reprotox or cancer, where there's an enormous number of possibilities. And that really, the skin sensitization, or ARE assay, takes advantage of some of that aspect of it. So just real briefly of how it works is we have a sensor protein called KEAP1. It's highly reactive cysteine residues. And when those bind covalently to these residues by electrophiles, the KEAP1, shown here, and NERV2, they dissociate from one another. The NERV2 translocates to the nucleus, activates these ARE response elements, and then it lights up target genes. So in this case, represented by luciferase. So this is genetically, a transgenic cell line. And so really the numbers so far coming out of this work, this is just one paper from a ring study that I think was done on 4 different labs, but they're showing predictivity, in the range of in the 80s and high 90s. Now it's only 28 chemicals, but there's other data sets out there that are all pretty much showing similar things. In our lab, we just started working on this a few months ago, but we're seeing similar encouraging kind of results. If you look across here, it's a comparison of the ARE. We also have an assay called the direct peptide reactivity assay. It's come out of Procter & Gamble. And then comparing that to some of the animal studies, the guinea pig test and the local lymph node assay in mice, and then human data. So the red's positives, the blues are negatives. And, reasonable concordance. I won't walk through all the details of it. But really the take-home message is not so much how did this ARE do against the other ones, but when we, when we combine the 2 assays, we're really seeing 100% concordance to the humans. And I think that is another take-home message, is that I don't think any in vitro assay is really ever going to get past maybe 80, 85% predictivity or overall accuracy by itself. But when you start to combine these things and all these different tools together, then you really start to move into the area where now you can start talking about using this to really make decisions that you're confident in. Cheminformatics, this is like the newbie toxicologist view of cheminformatics. It's kind of scary to compare to what you just saw in the last 2 talks. But essentially, what we're talking about is mining data on our chemicals or our candidate molecules, both structural analogs as well as substructures. Traditionally, we kind of do this now to some extent because, our toxicologists can go out to databases and mine them, but there's really not a lot of consistency to it, and just real exciting to see all the tools here that are becoming available. What the real interesting thing that has changed in recent years is the publicly available data. So some of the databases you're familiar with that came out of EPA computational talks, also some of the databases in Europe. There's mining tools out there to do all this. There's predictive models being developed all the time. And so this gives us an ability to do some really quick turnaround assessments that the people in R&D really will start to value now. So what are the limitations, though? Well, the levels of confidence really depend, as others have said in this today, is the availability of data on substructures and these substructures. So, and some of the areas we're starting to get into this. So already we're finding one roadblock is that the relevant data are our own data. They're not out in these publicly available data, because other companies work in different chemistries. So that's one issue which sometimes is a problem. The other one, it varies by endpoint, just like QSAR. There's QSARs are better for skin irritation and sensitization than they are for cancer or reprotox. I think the potential, though, here is if you combine this with the biological assay platforms, now you can start to get somewhere because some of the informatics can start to lead to testable hypotheses, and that's really exciting. So I think the other thing I think in terms of overall confidence is that while it might be not enough to base a lot of decisions on right now, it has the potential to greatly improve over time. The zebrafish embryo test, again, I was blown away by Dr. Tanguay's talk with that enormous facility. I know we've struggled just getting it up and running with trying to get these fish to spawn, but that's all working now. So, I know people used to say about rabbits too. Well, they breed like rabbits, not my rabbits, So, but I don't need to go through this assay again, but this is an assay that we're bringing on board. I really love the points that he made, though. The same reasons we like it is that it's an intact organism, really covers sort of a wide spectrum of possible effects. And it's very flexible. And I think for us to apply some of these new tools, I think in terms of platforms that are very flexible, I'm going to get calls from different businesses, R&D groups, and they're going to have a different issue every time. And I can't have, I can't have my own mini ToxCast at Dow. I have to have a platform that's flexible enough I can have the economy of scale, but still answer their questions. So one of our first projects really we're doing with some of our R&D folks is, again, more of a substitution kind of thing, where the issue is acute toxicity to fish. And so this is not our work, but a paper by Lammer et al., where they're comparing a simple thing of using the zebrafish embryo test and using that as a predictor of adult toxicity to fish. And you can see that the correlations are quite good. And this is over, so a 0.86 over 70 chemicals. Teratogenicity is certainly, we're looking at as a potential endpoint. I'd say, just like Dr. Tangway said, there's a lot of the predictions are quite good. I've seen, in fact, somewhere the zebrafish embryo test is doing a little bit better than, say, embryonic stem cells even. I also have seen in certain chemical groups, chemical space, where the predictions are pretty low. And I think that's always a common theme with any of these assays. It kind of works for certain chemistries and maybe not for others. So, and then the key issues are, I think, for this assay in particular, are just the test uptake by the kinetics, essentially. So I think there's some solutions to that, as he pointed out. And then ToxCast, I'm quite happy to say that we're collaborating with David and Tom Knutson's group at NCCT on a prototypical toxicity pathway, so that vascular development. And you did all the hard work for me by showing all those nice slides, so I don't really have to describe that. But our general questions for this toxicity pathway is, do the signals in ToxCast really correlate with more functional endpoints? So as David showed you, the EPA's part is they have the ToxCast assays, and there's a subset that you can do, say, a ToxPi score for vascular disruption. And then also the computer simulations, which he actually showed. And then for our part, we're doing more functional assays, still in vitro. So aortic explants, looking at angiogenesis, and the whole embryo culture model, which we've had in my lab for a long time. And we're using these thalidomide analogs, again, as positive controls. So the exciting thing is, I don't really have too much to show you on that with real data, but the exciting thing is on these computer simulations that have been done are, let me go to the next slide. So if you did a close-up on these are the explants. So you take a piece of aorta and these little vessels start to form out over a period of about a week. And you can see the inhibition that's occurring here. But if you did a close-up of some of the morphology here, it's starting to look really a lot like these computer simulations, which that kind of blows me away, really. It's really exciting. And then these are the results we're seeing in the embryo culture. So control embryo, and you can see that it gets a little smaller here, and then we start to lose some of the caudal structures in particular are affected. So real early data, but to me it's kind of encouraging that we're seeing some correspondence across these 4 different really platforms. And so really, here's my one little bit of cautious advice on this whole thing is, where do we want to I think we have to build confidence over time and just sort of face some of the tough issues and deal with them square on. And our approach really has been to start with the things that we have the highest confidence in right now, so things like skin sensitization, and sort of build out from there. The publicly available databases, I think I mentioned some of the limitations, that it's not, some of the chemistry may or may not be relevant, but overall, this is a huge resource. It's almost like EPA and some of the agencies that are putting these data out there are now kind of part of our R&D. And I think we've just never been there in industry before, and I think, to me, that's super exciting. Building the knowledge bases, I think I beat that one to death, but I think one part, I think, for me, a reality of all this is as we sort of build this whole new program, there probably will be need to do some targeted in vivo testing as well to do the phenotypic anchoring. I don't think we can really get around that. But I think I'm not talking about doing standard guideline tests when I say that. I'm talking about targeted research. So it should use a lot fewer animals. But, we don't want to kind of sacrifice the current levels of confidence that we have. I think that is, as much as we're excited about all this, it could backfire on us if we use these decisions too early, before they're not ready, and sort of blow off some of the limitations. If you think of my slide with the canyon, you can kill yourself on either end if the bridge breaks on the left side or the right side. And so if you sever the tie to the traditional too early, that's could really be a problem for this. So, let's do this in a stepwise fashion. But bottom line is, let's be optimists and try to imagine the possibilities. I think it's really exciting to be part of this. So just a few acknowledgments, a few folks in our lab, some of the names you might recognize at Dow, our collaborators, especially Tom and Nicole and David in the audience, Andreas from Givaudan, and George Dasson, who's wearing a green shirt today. He's been really helping me with a lot of the thought. And we're collaborating with George's group, where I didn't get into that too, but that's very exciting. And finally, for Martin for giving me the call to invite me to give this talk. It's a great honor. Well, thanks.
We'll take a couple of questions. So Ed, thank you very much. I think it was exactly the right balance of excitement and criticism. As I was listening to you, I couldn't help but think of that you're, well, actually, a little bit putting my words into your mouth, almost like, built for purpose in vitro testing, which is good in a way because it increases confidence, but I think it's a limitation because there's always going to be a criticism of, my favorite toxicity or this thing that is very, very important, and some of the more complex one like neurotox or something else. That we don't have a good assay for that purpose, and hence we will never be able to untie that knot to the traditional toxicology.
Right.
So what do you say, to your colleagues, who may have this argument?
No, that's a great question. It's kind of a fundamental question. And again, I sort of step back to what we have now, the so-called traditional tox. That's sort of ripe for criticism, too. I think one of the frustrations I've always had as a practitioner in developmental tox is even when you do a 2-gen study, which costs us $500,000 and all this effort, uses over 3,000 animals, and you still get criticism, well, what about this? What about that? So that's where we are now. So, we have that. I think the path forward is kind of a combination of the assays like the sensitization again that is pretty anchored to a very discrete endpoint, and so those things I think we'll get the rapid successes there. But on the other end, the more complex, your cancer and your repro, your neurotox, I think a fundamentally different approach is really ultimately what you're going to need, because you have to really cast a wide net. And so you look to what technologies that cast a wide net. I think to omics for sure casts extremely wide net. To genes we don't even know what they do, right? ESTs and stuff. So I think that is a longer ways out, but I think that's, to me, demands a newer approach.
Thank you. We can only take one question from a reproductive toxicologist.
Excellent. Oh,
no, George, I'm joking.
No, I used to be.
David, go ahead.
So, David Dick, CPA. Ed, that was a nice presentation. I got a little excited. I thought you were going to show an analysis of ToxCast data on DynaCap. But maybe tomorrow, maybe later today, but maybe by tomorrow I can... Because one of the things, and you use the cast a wide net, which is the metaphor for ToxCast, of throwing a broad net.
Right.
And one of the things that you can find potentially from a genomics data set or another omics data set, or maybe from the ToxCast data set, is some surprising associations of molecular targets or cellular targets that may only have a statistical association with an endpoint like you showed for DynaCap, but there might be some some sort of discovery biology, some underlying biological plausibility that we just don't appreciate at this moment. So it's something we can look forward to as we continue to build these HTS data sets, I think.
I
so
totally
not really
agree.
A question unless...
Yeah, it's our comment. Yeah. No, I agree, though.
Unless you could come up with another metaphor for the bridges. I was thinking about a sinking ship on the one side and a gangplank back to shore, but
Yeah, no, I totally
no
agree.
questions.
Because you only know what you know, right? And so that's the value of some of this stuff, is to generate new hypotheses.
Yeah.
Some may be dead ends, but some of them will lead somewhere. Yeah.
George?
George Dasson, Procter & Gamble. So maybe this would be better for the discussion section, but one of your slides kind of prompted me, which is in the chemi-informatics slide. What I really liked about that slide is it ends in a toxicologist or a toxicology assessment. And I think, I was thinking about this as Alex gave his talk, which is, one of the real problems in the development of in silico models has been the quality of curation or the lack thereof. And I actually, because of that, of all the data streams that one can start with in a predictive model, we pretty much settled on a chemical analog approach first, because it immediately forces you into an expert assessment of the quality of the data on which you might base your other assessments. And I'm just wondering, in your opinion, do we have the kinds of data streams? I mean, you work for a big chemical company that makes lots of lots of different kinds of things. I mean, are the data out there to support those kinds of expert-based judgments based on analogs?
Yeah,
can we start
you should have
there?
been in the meeting room a week ago. We had a passionate discussion about this internally, and I think it varies. The one opinion was the one I expressed is that this is all great, but when we look at our chemistries, our analogs are our chemistries, so they're not really out there. But I think in other areas they are. I can think of in the agricultural space, there's we're especially, I can think of one compound which is has a very discrete insecticidal target, and there's competitor molecules in the same space. There's some pretty good data out there, and having toxicas data on those chemicals could be pretty useful. So, I guess that's the extent of my answer, but I think it'll build over time. But
well, let's thank Ed and the rest of the speakers in this morning's and afternoon session.